Good afternoon, everybody. I'm very happy and I thank the organizers to give me the opportunity to share the results of the study. As you can see, I think I'm the only not Italian here. I am sorry. So I really will do this presentation in English because otherwise it will just not be possible in Italian. I was asked to present the results of this study. It's a prospective interventional study with the ASTar approach on 200 patients with gram-negative blood cultures. I will share with you the microbiological performances, but of course also the clinical impact that we were able to measure on these 200 patients. This is an academic study, it's not a commercial study, so the study was designed and the results were analyzed by the hospital team without any input from Q-linea. So what were the objectives of this study? Of course, first of all, our aim was to measure the microbiological performances. The reference method that we used was subculture broth microdilution, which is also considered as the gold standard when you start to compare AST results. We analyzed all results with the UCAS 2021 breakpoints. These are the breakpoints that also include the new I, susceptible high exposure, which made it sometimes a little complicated to measure the very major errors, the major errors, and the minor errors. So you know for all antibiotics for which a susceptibility category was missing, where there was only an I and an R category, we considered very major errors and major errors if there was a discordance and not minor errors. We also evaluated the ease of use of the system. You will see we used the system for a little more than 6 months. The hands-on time, the reagents, also the system failures, and of course the time savings towards AST results. Then we measured the clinical outcomes, which is probably what we all want to know. So the impact of the communicated ASTar result to the clinician on the antibiotic tailoring of the patients with positive blood cultures. And also we measured the time savings that were gained due to the earlier administration of the targeted antibiotic treatment. So this study is a monocentric study that was performed at the Cliniques universitaires Saint-Luc. It's a very big hospital, in the center of Brussels. Our microbiology laboratory is working 24 hours out of 24 for blood culture incubation, but considering positive blood culture management, it's performed from 7 in the morning to 9 in the evening. So all the blood cultures, unfortunately, that get positive during the night are only managed the morning after. So we were able to include prospectively 200 adult patients with a routine gram-negative detected blood culture during weekdays. The study was performed from Monday to Friday, and we always included the first positive blood culture of each bacteremic episode. What were the exclusion criteria? At the time that the blood culture was detected positive, the patient was excluded if he deceased, if he was transferred by that time to another hospital, or if by then the decision was taken that the patient was transferred to palliative care. So this is the workflow, the way we manage our positive blood cultures. In blue, you have the routine workflow, and in green, you have the study workflow. So the study started in November 2022, and we were able to include 200 patients by the 2nd of June 2023. So we included all patients, adults with positive detected blood cultures with a negative gram stain. What do we do in our routine? For the identification, we do MALDI-TOF, either on very young subcultured colonies, either on blood, which allows us to have the identification result on the day of positivity detection. Considering AST, we actually have two tests. We have a rapid resistance detection test that allows us, it's a colorimetric test that allows us within 15 minutes to identify whether the strain produces an ESBL or not. That's also done on day 0, so you have the result on the day the blood culture is detected positive. And we also do the classical automated AST on young subcultured colonies, so we have the results the day after positive blood culture detection. And then the study part, well, then you can see we have the ASTar approach. So you know it was actually a quite big study because we also challenged 2 additional RAST approaches at the same time. So we initiated as soon as possible, as soon as we had the gram stain results, these RAST approaches on weekdays between 8 a.m. and 6 p.m. The earlier we started the test, the earlier we had the result. And when the test was started in the morning, the result was available in the afternoon. When the test was started in the afternoon, the result was available during the nighttime, but that was only communicated the day after. Very practically, what did we do with the results that we obtained? So once the RAST result was available, we called, the microbiologist called the infectious disease physician. This is something we always do. Every afternoon, we call the infectious disease physician to communicate them the results, the new results of the positive blood cultures. So we call them in the afternoon and we discuss the antibiotherapy that the patient is currently receiving. Then we communicate the MALDI-TOF identification result and the result of the rapid resistance detection test. That's what's already done in our routine, and we discuss if these results enable the clinician to modify, to target even better the treatment. But in this study, we communicated during the same phone call the ASTar results, and then we discussed again. So the thing was, okay, now that I give you the additional ASTar results, like all the antibiotics and all the results, is this also going to change something to the therapy that you chose for your patient? And if the answer was yes, then that change was truly made, and we measured the time, the reduced time of the administration of the targeted treatment. So let's go to the results. Like I said, we included 200 patients with gram-negative blood cultures. Ultimately, of those, we had 7 contaminations. So we got back to 193 bloodstream infections. Of those, we had 5 gram-negative bloodstream infections that we discarded because you cannot do an AST on a polymicrobial blood culture. So we have left over 188 monomicrobial bloodstream infections. Of those, you see the details: 158 enterobacterales, 17 non-fermenters, 2 Haemophilus influenzae, 2 Echinella corrodens, and 9 anaerobes. On the next slide, you see the details of all the identifications of those 188 bloodstream infections and also the resistance profile. So I already hear you thinking, this is not like in Italy, and I completely agree. Our resistant epidemiology is not the same. In our blood cultures, we approximately have 10% ESBL, and carbaphenemases are very rare. Actually, we felt lucky, if I can say it like that, to have 2 carbaphenemases in this 6-month period. So you see the details of the identifications. Those in red are currently identifications that are not leading to ASTar results. So these are not in the software yet, and they don't enable any AST results. So this is something you have to be aware of. So ultimately, of the 188 monomicrobial bloodstream infections, ASTar enabled 163 AST results, which is approximately 87%. The microbiological performances on these 163 evaluable ASTar tests, you see them here. I divided them for you in 3 groups. We look first at the Enterobacterales, 146 positive blood cultures, a very nice categorical concordance with subculture microdilution reaching 97%, 2.6% very major errors, 1.8% major errors, and 1.3% minor errors. What we also analyzed is that we had a closer look to all those errors that we had, and we looked if these errors were on the edge of a category, if they were on the edge of S versus I or I versus R, and if they were in an essential agreement. And as you can see, of all the errors, 90 errors, actually more than half of them were in an essential agreement. When we look at the non-fermenters, we were able to evaluate 160 strain drug combinations, excellent categorical concordance of 99.4%, only one very major error for levofloxacin. And then we had 2 Haemophilus influenza, so we cannot really draw any conclusions, but for those 2, all results were completely concordant with routine testing. Now some information about the time analysis, because this is rapid AST, so we really want to see that it's rapid and that it has some consequences on the patient. So what did we measure? All our time measurements started at the time of positive blood culture detection, because it's from that time that we, as microbiologists, can make a change. So we had a median time of 9 hours and 48 minutes to MALDI-TOF identification. That's our routine approach. We have a median time of 10 hours and 12 minutes towards the result of the rapid resistance detection test. And then when we included in this workflow the ASTar AST, well, we were able to communicate to the clinician the ASTar AST results 12 hours after the detection, a median time of 12 hours after the detection of the positive blood culture, which is a major gain compared to routine AST that we already do on young subcultured colonies. So we already tried to win some time there, but still the median time of routine AST in our management is 30 hours and 34 minutes. So with ASTar, we have a time gain of 18 hours and 36 minutes on AST performances. Considering the ease of use in the system, well, hands-on time is very short. It's 1 minute. It's extremely easy. We did have 2 system failures over the 6 months. But to be honest, we called each time and the problem got resolved very quickly. Within the day, the problem got resolved. Considering the reagents, also something important, the reagents are stored, there are 2 reagents, one is stored at room temperature and one is stored at the freezer. The clinical outcomes, probably the most interesting results. First, some global data considering the patient distribution. We have a very correct distribution between males and females. Median age of all these patients that we included, 66 years. The main source of the bloodstream infections that we included was the urinary tract, followed by the gastrointestinal tract, followed by febrile neutropenia and the respiratory tract. Where did these patients come from? Most of them from internal medicine, followed by 17% from surgery, followed by intensive care, 16% of the patients. And this is probably the most interesting slide of this presentation. Here you really see the evolution of the antimicrobial treatment following the modifications based on the communication of the microbiological results. I explain myself. First here you have the empirical treatment. This is the treatment that the clinician chooses to give to the patient before he gets any results of the lab. It's really, I think my patient has a bloodstream infection, I need to give him something, and so this is the empirical treatment. In our setting during this study, almost 50% of the empirical treatments were ultimately also the targeted treatment. So right away, we already had 50% of the patients receiving the appropriate treatment. But also, 23% of the choices were choices that were completely ineffective. So we have there a large group of patients that receive an ineffective empirical treatment. And then we have 26.6% where the chosen treatment had a too broad spectrum. Then, following the results of the MALDI-TOF identification and the rapid resistance detection test, things improved. We had an increase of the targeted treatment and, of course, a reduction of the ineffective and the broad spectrum treatments. And then remember, in the same phone call, we also discussed the ASTar results that we additionally communicated. And then again, we said, okay, now that I give you this information, are you going to do some additional changes, or are you keeping it like you decided before the ASTar results? But there have been approximately 15% additional changes due to the communication of the ASTar results, which goes with a major reduction of the percentage of patients with an ineffective treatment or a broad spectrum treatment. And then there were still a 6% additional patients that only received the appropriate, effective, targeted treatment once the routine AST treatment was communicated. How we explain these 6%? Because it's a study, it's something that requires trust from the clinician, and there has been a very few situations where the clinician really said, okay, but yes, your system is new, I'm not going to change anything, I just want to wait the results from your routine AST. So this is also very important. It's a question of trust, and it takes really time to work with the clinicians and to convince them of the appropriateness of these new technologies. So ASTar results, when we look at the clinical history of these 200 patients, enabled 27 antibiotic modifications. So we could say, okay, we have an impact of 27 out of 188 monomicrobial gram-negative bloodstream infections, which is 14.4%. 33% narrowing, 55% broadening, and 11% switch. These patients truly benefited from a huge reduced time towards the administration of the appropriate targeted antibiotic treatment with a gain of 20 hours. Now, if I look at these percentages, 14.4%, I'm thinking, okay, nice, but can we not do better? This is like a big change, an investment. I really want this to work. I want this to be useful as much as possible for the patients that I choose to do an ASTar test on. So how can we explain these 14.4%? First of all, I really believe there's a bias due to the weak local resistance epidemiology, which is good to have not that much resistance, but as you can see, this also means that the appropriate treatment is already good. 50% of the empirical treatment is already the appropriate treatment. We also have very strong stewardship guidelines, which really helped in the selection of the appropriate empirical treatment. I also believe that the rapid resistance detection test in our setting is very useful. As I said, we have 10% ESBL. That's our main resistance in Enterobacterales. So it really helps the clinician to modify the treatment upon the availability of that rapid resistance detection test. Finally, I think there was also a bias in the study design. By the end of the study, when I had a clinician on the phone and I communicated to him the results of identification and the rapid tests, he knew that 2 minutes after I was going to communicate to him the ASTar results. So he knew if he made a change, he would be able to correct it upon the ASTar results. So it was time that the study finished and that we could put the system in routine to do it correctly. And then finally, we had some thoughts about the study workflow. Like I said, the tests were started during the entire day. For example, we did start some tests in the late afternoon. This means that the results were available during the night. In our setting, like I said, unfortunately, there is nothing going on during the night. So even though the results were available at midnight, we were only able to communicate them in the early morning the day after. So you really lose the added value of these rapid testing if you don't act upon it right upon the availability of the results. So we have been thinking, and after this study, how we could improve that percentage of impact of our ASTar approach. And there was, according to us, really a necessity of adapting the blood culture workflow in our lab, which is not always easy when you have to make changes. It bothers sometimes people. A first thing is currently we have to put a correct time slot, a useful time slot for RAS testing, which is in our lab during the morning, until we are able to communicate the results during the night and until we are able to have some clinicians that are willing to listen to the results during the night. I think it's also important, and specifically for intensive care, who is awake 24 hours out of 24, to enable a result transmission towards the patient's file prior to medical validation. As you remember, we also had some strains that, for which no ASTar result was available because these Enterobacterales are not reported yet, or not covered yet by the system, and we also had many anaerobes. Maybe it's also a thought that we first should do the identification and then decide if we do or we do not a RAS test. If we apply all these criteria on our study population, we would certainly be able to improve the impact of the RAS results towards almost 25%, 24%, so one patient out of 4, which would really be a huge improvement. So, considering other points, what we found, our team found of the system, it was very easy to use. The panel of antibiotics is very large. We have some small spectrum antibiotics, which we like to use, cefuroxime, ampicillin, ceftriaxone, but also you have very broad spectrum antibiotics that can be used. We had 2 system failures, but they were rapidly resolved, and there are only 2 reagents. What I missed is the colistin for several Enterobacterales, and for non-fermenters, this is not available yet on the system. So I would stop here, but then again, I know I'm in Italy and that you have some more resistances than we do. And so it's important also to challenge the system with multi-resistant gram-negative bacteria, because this is probably our future also, unfortunately, in our country. So we did a study, a microbiological study, where we tested 42 positive blood cultures on the system, blood cultures that we spiked with multi-resistant bacteria. Here you see the details. We had 32 Enterobacterales, half of them were ESBL and half of them were carbapenemase producers, and we also had a gram-negative non-fermenters, all with a carbapenemase. Testing was done in the same way as the clinical study, and here you see the performances of on these 42 spiked blood cultures. For the Enterobacterales, we had a categorical concordance of 94.2%, with 2.8 very major errors, 2.9 major errors, and 3% minor errors. But then again, when we had a close look to those categorical errors, we observed that almost 42% of them were in essential agreement. And considering the non-fermenters, we had a categorical concordance of 96.5%, no very major errors, mainly major errors that all were corrected when we measured the essential agreement. So as a conclusion, this study was very helpful for us to draw some conclusions on, okay, is this really useful for the patient? Okay, the microbiological performances are very good, but can I improve the management of the patient? The answer is yes. But then again, I already hear my medical director saying, okay, Alexia, you want to buy the system? Well, you just have to prove me an impact on mortality and on length of stay. We always get there. Unfortunately, studies are lacking. These are very new technologies, so studies are really needed to show that. What I show you here is a study of the effect of rapid identification on positive blood cultures. And this is a review with 5,920 patients. And when you already see how big the impact is of rapid identification in positive blood cultures, as it prevents 1 death within 30 days, as it reduces the total length of stay with 2.5 days, we can only imagine the major impact of rapid AST on length of stay and on mortality. And the next question would be, okay, great, but now let's talk money, because that's also something important. Well, we need health economic studies, and maybe this helps you to introduce the next speaker, who certainly will give you some insight about that. I thank you for your attention.